Dear students, today's practical class is on the determination of glucose and an amino acid. Determination of glucose by Nelson-Somogyi method. First, the principle. The method is based on the reducing property of sugar and is again based upon the fact that in solution the reducing sugar passes a free aldehyde group and is capable of reducing an alkaline solution of cupric ions. When glucose is heated with alkaline copper tartrate, the cupric ions in the solution are reduced to cuprous ions and finally cuprous oxide with red precipitate. Cuprous oxide reacts with arsenomolybdate reagent which reduces arsenomolybdate to molybdenum blue. Its extinction is red at 620 nanometer. The amount of color produced depends on the amount of sugar present. Thus, the reducing sugar can be quantitatively assayed by this method. Reducing sugar plus alkaline copper tartrate on heating gives cuprous oxide. Cuprous oxide again reacts with arsenomolybdate to give molybdenum blue. Now let us see the requirements. Reagents. Preparation of alkaline copper tartrate solution. Solution A, 1.25 gram of anhydrous sodium carbonate, 1 gram of sodium bicarbonate, 1.25 gram of sodium potassium tartarate and 10 gram of anhydrous sodium sulfate were altogether dissolved in 40 ml of distilled water, and finally the volume was made up to 50 ml with distilled water. Next, the solution B. 1.5 gram of copper sulfate is dissolved in small volume of distilled water and a drop of concentrated sulfuric acid is added. The volume is made up to 10 ml with distilled water. 2 ml of the solution B is mixed with 48 ml of solution A just before the use in the experiment. Next, the preparation of arsenomolybdate solution. 2.5 gram of ammonium molybdate is dissolved in 45 ml of distilled water. 25 ml concentrated sulfuric acid is added to it and mixed well. 0.3 gram of disodium hydrogen arsenate is dissolved in 25 ml of distilled water, mixed well and incubated at 37 degrees Celsius for 24 hours. The solution is not allowed to expose to light. Next, preparation of standard glucose solution. Standard stock solution is prepared by dissolving 100 mg of glucose in 100 ml of distilled water to give 1 mg per ml. The working standard solution is prepared by diluting 1 ml of the stock solution with 9 ml of distilled water. The concentration of the standard working solution is 100 microgram per ml. Coming to the procedure. Aliquots of the standard glucose solution 100 microgram per ml are pipetted out in a series of test tubes as 0.2 ml, 0.4 ml, 0.6 ml, 0.8 ml and 1 ml. The volumes of each test tube is made up to 2 ml with distilled water. Then, a blank is also prepared containing 2 ml of distilled water. 1 ml of alkaline copper tartrate solution is added to each tube including the blank. The tubes are covered with marble and kept in boiling water bath and then cooled. 1 ml of arsenomolybdate solution is then added to all the tubes. Finally, 6 ml of the distilled water was added to each tube and shaken well. Extinction at 620 nanometer using red filter in the spectrophotometer was read for all the tubes against the blank. A standard plot was drawn with the concentration of the reducing sugar on the x-axis and absorbance at 620 nanometer on y-axis. The test tubes are assigned 1 to 9. In the blank test tube, no working standard solution is added and only distilled water 2 ml is added. In the second test tube, 0.2 ml of working standard solution and 1.8 ml of distilled water is added. In the third test tube, 0.4 ml of working standard solution is added and 1.6 ml of distilled water. In the fourth test tube, 0.6 ml of working standard solution is added and 1.4 ml of distilled water. In the fifth test tube, 0.8 ml of working standard solution and 1.2 ml of distilled water is added. In the sixth test tube, 1 ml of the standard working solution and 1 ml of distilled water is added. In the 7th, 8th and 9th test tube, 2 ml of the unknown samples U1, U2, U3 is taken and no distilled water is added. To the following test tubes, 1 ml of alkaline tartarate solution is added. All the test tubes are incubated in boiling water bath for 10 minutes and cooled at room temperature. After that, 1 ml of arsenomolybdate solution is added. Then, 6 ml of distilled water is added to all the test tubes and is kept for 10 minutes at room temperature. The absorbance of the test tubes are read against 620 nanometer. The blank reading gives 0.00. The second test tube gives 0.16. The third test tube gives 0.28. The fourth test tube gives 0.32 reading. The fifth test tube gives 0.60 reading. The sixth test tube gives 0.70 reading. The seventh test tube gives 0.28 reading. The eighth test tube gives 0.30 reading. And the ninth test tube gives 0.32 reading. Observation. The concentration of the unknown sample can be deduced from the standard plot. The given unknown sample contains 22 microgram per ml of glucose. The second experiment is the amino acid estimation. Estimation of amino acid by colorimetric method using ninhydrin. First, the introduction. The amino acids found in proteins arise mainly from the digestion of dietary proteins. Some amino acids can be synthesized by the animal, and these are known as non-essential amino acids, while others, the essential amino acids, must be supplied in the diet. The alpha amino acids, as well being concerned in protein synthesis, are also involved in the synthesis of a number of compounds of biological importance. Coming to the principle, ninhydrin, that is triketohydrindin, is a powerful oxidizing agent. It can react with alpha amino acids in between pH 4 and 8 and bring about oxidative decarboxylation of the alpha amino acids, producing carbon dioxide, ammonia and aldehyde with one less carbon atom than the parent amino acid. The reduced ninhydrin, hydrindentin, then reacts with the liberated ammonia and another molecule of ninhydrin to form a purple colored complex called women's purple, which is colorimetrically measured at 570 nanometer lambda max. Not all amino acids gives a purple color complex. Proline and hydroxyproline give a yellow color, so these are read at 440 nanometer. Coming to the requirements, concentrated hydrochloric acid, aspartic acid, weighing balance, pipettes, spatula, volumetric flask, sodium acetate, acetic acid, test tubes, beakers, acetone, 50% ethanol, water bath, distilled water, spectrophotometer, filter paper, tissue paper. Now let us see the procedure. First, preparation of standard solution of aspartic acid. 100 ml of 0.01 normal HCl solution is taken. To it, 0.005 gram of aspartic acid is added. The solution thus prepared is a standard solution of aspartic acid of 0.05 mg per ml. Next one, 0.2 molar acetate buffer pH 5.5. Preparation of 0.2 molar acetic acid solution. 1.15 ml of acetic acid is taken and the volume is made up to 100 ml with distilled water. The acetic acid solution thus prepared is 0.2 molar acetic acid solution. Next, preparation of 0.2 molar sodium acetate solution. 1.65 gram of sodium acetate is dissolved in 100 ml of distilled water. The solution thus prepared is 0.2 molar sodium acetate solution. Next, preparation of 0.2 molar acetate buffer of pH 5.5. 84.6 ml of sodium acetate solution and 15.4 ml of acetic acid solution are mixed. The prepared solution is 0.2 molar acetate buffer of pH 5.5. Next, preparation of ninhydrin reagent. 4.8 gram of ninhydrin is dissolved in 60 ml of acetone. To it, 60 ml of 0.2 molar acetate buffer is added. The solution thus prepared is the ninhydrin reagent. Now coming to the procedure. 6 test tubes are taken and to it 0 ml, 0.4 ml, 0.8 ml, 1.2 ml, 1.6 ml and 2 ml of standard aspartic acid solution are added respectively. 2 ml of the unknown sample is taken in a test tube. The volumes of the test tubes are made up to a total volume of 4 ml with distilled water. Then 1 ml of ninhydrin reagent is added to each of the test tubes. The test tubes are then incubated in a boiling water bath for 15 minutes and it is then cooled at room temperature. After cooling, 1 ml of 50% ethanol is again added to each of the test tubes. It is then incubated for 10 minutes at room temperature. Absorbance of each test tube is then measured at 570 nanometer. The absorbance is noted down and a graph is drawn by plotting the absorbance against their respective aspartic acid concentrations. 7 test tubes are taken and assigned as 1, 2, 3, 4, 5, 6 and 7. In test tube number 1, no aspartic acid solution is added and only 4 ml water is added. In test tube number 2, 0.4 ml of standard aspartic acid solution and 3.6 ml of water is added. In test tube number 3, 0.8 ml of aspartic acid solution and 3.2 ml of distilled water is added. In test tube number 4, 1.2 ml of aspartic acid solution and 2.8 ml of distilled water is added. Test tube number 5, 1.6 ml of aspartic acid solution and 2.4 ml of distilled water is added. In test tube number 6, 2 ml of aspartic acid solution and 2 ml of distilled water is added. In the 7th test tube, 2 ml of the unknown sample and 2 ml of distilled water is added. The amount of aspartic acid in microgram in the respective tubes are 0, 20 microgram, 40 microgram, 60 microgram, 80 microgram and 100 micrograms. To all the test tubes, 1 ml of ninhydrin reagent is added and then incubated in water bath for 15 minutes and then cooled at room temperature. To this, 1 ml of 50% ethanol is added. The total volume of each test tubes is 6 ml. The tubes are then incubated at room temperature for 10 minutes. The absorbance of the tubes are read at 570 nanometer. The test tube number 1 gives 0 reading. Test tube number 2 gives 0.212 reading. Test tube number 3 gives 0.421 reading. Test tube number 5 gives 0.689 reading. Test tube number 6 gives 0.84 reading. Test tube number 6 gives 1.311 reading. And test tube number 7, that is the unknown test tube, gives 0.631 reading. Now coming to the observation. The amount of amino acid in microgram is plotted in the x-axis and the absorbance at 570 nanometer in the y-axis. Now coming to the conclusion. The ninhydrin reaction, one of the most important methods of detecting amino acids, both technically and historically, has been conventionally used to detect their microgram amounts. When amino acids with free alpha amino groups are treated with an excess of ninhydrin, they yield a purple-colored product. Under appropriate conditions, the color intensity produced is proportional to the amino acid concentration. Nelson-Somogyi method is the most used one for the quantitative estimation of reducing sugar, as it has a high degree of stability on keeping, whilst the high content of the sodium sulfate and absence of iodide prevent reoxidation of the cuprous oxide formed. Thank you.